All right, so we are now at the second topic of the fourth unit, which is now all about protein. So we will now study the different hierarchy of protein structure. It starts with the primary structure, followed by the secondary structure, tertiary structure, and the quaternary structure. As you can see here, as the structure increases, or as the hierarchy increases, it becomes more complicated. We now first discuss the first structure or the primary structure. So the primary structure is about the amino acid sequence. So it contains all the amino acid sequence. The linear order of amino acids from the N terminus to the C terminus. It dictates the structure and function of the protein. So as you can see here, an example of a primary structure coming from the arginine, proline, aspartic acid, phenylalanine, methionine, isoleucine, alanine, and valine. So this is an example of of an amino acid sequence. In your exercise, in your book, um, a question there is all about what happens if a sequence in the amino acid sequence is being altered or varied. What will be its effect on the structure or also in the function of the protein? So one good example is the sickle cell hemoglobin. So here is a table which shows the difference or comparison of a normal hemoglobin and a sickle cell hemoglobin in terms of their structure, their function, and their shape. So we have here the normal hemoglobin in terms of the primary structure. So this is the normal hemoglobin structure. When it is already a sickle-celled hemoglobin, the glutamic acid or the sixth amino acid becomes valine. For the secondary and tertiary structures, a normal hemoglobin looks like this, while a sickle cell hemoglobin looks like this. So there is an exposed hydrophobic region. For quaternary structure, the normal hemoglobin looks like this, and the sickle cell hemoglobin looks like this. In terms of function, molecules do not associate with one another in a normal hemoglobin, so each carries oxygen. While the sickle cell hemoglobin, the molecules interact with one another and crystallize into a fiber. Capacity to carry oxygen is greatly reduced. For its shape, so the red blood cell shape, in the normal hemoglobin, it is full of individual hemoglobin molecules and each carrying oxygen. However, in the sickle cell hemoglobin, the fibers of the abnormal hemoglobin deform red blood cell into a sickle shape. That's why it's called a sickle cell hemoglobin. It is sickle shaped already. Now we go to the secondary structure of proteins. So it is the spatial arrangement of amino acid residues that are near one another in the linear sequence. Common regular folding patterns of the polypeptide backbone. So the hydrogen bonds between peptide bonds is the main interaction. Secondary structures include alpha helix, beta pleated sheets, and collagen helix. So let's discuss this one by one. So the alpha helix, so look at this, it's a helix structure. A polypeptide backbone is tightly wound around an imaginary axis that is drawn longitudinally in the middle of the helix. So this is the alpha helix. You can see there that is a helix structure. The next one is the beta pleated sheet. So the backbone of the polypeptide chain is extended into a zigzag rather than a helix. The zigzag polypeptide chains are lying side by side and held together by hydrogen bonds. So these yellow lines here are the hydrogen bonds. So this is a zigzag structure. Pleated due to alternating position of the alpha carbon. So there is an alternate position of the alpha carbon. The next structure is the tertiary structure. So basically, it is the overall 3D or three-dimensional arrangements of all atoms in the protein. So you can see here an example. Results from folding into specific 3D shapes and unique binding sites. This is the highest level of protein architecture common to all biologically active proteins. Tertiary structure. So here are some of the facts about this. So the first one is that hydrophobic interactions occur in the tertiary structure. There is an overall tendency of side chains of nonpolar residues to collect in the interior of the protein. There is also a hydrogen bonding between side groups or between peptide bonds. There is also an electrostatic interaction between charged side chains. And lastly, there can occur disulfide bonds or bridges that are formed by two cysteine residues. So this is what we call the disulfide bonds. So that is actually a covalent bond between two cysteine residues. You can see here. Last one is the quaternary structure. So this describes the organization of subunits of polypeptide chains with tertiary structure in a protein with multiple subunits or multimeric. Subunits are held together by non-covalent bonds. It can have a homomultimers or heteromultimers. So this is how a quaternary structure of a hemoglobin molecule looks like. So you can see there, so this is the red blood cell. So this is a combination of four polypeptide chains and a heme group. You can see there. There is a heme group and the four polypeptide chains. Now we go to the protein folding. So protein folding refers to the folding of the protein, which gives its three-dimensional shape, which is critical in the function of that protein. So it is the manner by which proteins fold is by its amino acid sequence. Proteins fold into specific conformation that results in minimized free energy of the molecule, wherein the molecule becomes more stable. So here is an example of a protein folding from the primary structure to the secondary structure until the tertiary structure and finally the quaternary structure. So in an aqueous environment, the polypeptide chain folds in such a way wherein hydrophobic side chains are buried in the interior and polar side chains are on the surface. So you can see here, based on the legend or in the color, the yellow group or the hydrophobic groups are buried in the interior because they are hydrophobic, meaning they don't like water, while the polar side chains are on the surface. So these are the blue color or blue molecules in there. So those are the hydrophilic groups. They are on the surface. So here is the difference between an unfolded polypeptide and a folded conformation in an aqueous environment. As you can see, the hydrophobic core region contains the nonpolar side chain, as what we've discussed a while ago. And there is hydrogen bond that can be formed to the polar side chains on the outside of the molecule. So what happens when there are misfolded proteins? Misfolded proteins must be targeted for degradation because if there is an accumulation of these misfolded proteins, it can cause some diseases such as Alzheimer's disease, Parkinson's disease, mad cow, or senile dementia. Now we go to the protein denaturation or protein denaturation. So it refers to the alteration of the secondary, tertiary, and quaternary structure of proteins. As you can see here, this is a normal protein. And after denaturation, it becomes a denaturated protein, which looks like this. So the primary structure of the protein is preserved in peptide bonds is not easily disrupted. In protein denaturation, it may result into reduction or complete loss of biological activity of the protein, accomplished by the following agents of denaturation. So we have two agents of denaturation. We have the physical agents and the chemical agents. So the physical agents include heat and pH extremity. So also chemical agents include strong acids and bases, heavy metal cations like lead and mercury, alkaloidal reagents, and organic solvents like ethanol and acetone. Lastly, we now go to the classification of proteins in terms of their biological function. So we have here seven, but we will include, of course, but the nutrient and storage proteins in your book is separated, so it becomes eight. So we will discuss this one by one. So the first one is the enzyme or the enzymes. These are proteins that can catalyze biological reactions. Example of which are the lipase, amylase, pepsin, and trypsin. On the other hand, transport proteins function as they bind or carry specific molecules or ions from one organ to another. So examples of which are albumin and hemoglobin. We now go to the next class of albumin. So the function is to mobilize by the body to derive building blocks or energy. The next one are the defense proteins or or those proteins that protect. So they defend organisms against invasion by foreign substances. So example of which is the immunoglobulin. The next one is the contractile or motile proteins. This is for the movement. So it allows cells and organisms to contract, change shape, or move about. So examples of which are actin and myosin. The next one is the structural protein. This acts as supporting filaments, cables, or sheets for strength and protection of biological structures. Examples of which are collagen, elastin, keratin, tubulin, and actin. And the last one is the regulatory proteins, which acts as regulator for physical or cellular activities. Example of which is the ubiquitin.